everyone. Welcome to our masterclass today. My name is Nishika Sabharwal, and I'm the part of research for protein purification at Cube Biotech. And I'm responsible for protein purification, optimization, screening, and assay development. Today, I'm really honored to host this Intelligent Infusion Masterclass from Cube Biotech, created for all the researchers to give them the opportunity and a platform to share and promote their research with a worldwide audience. We are so grateful and happy to introduce our speaker today, Professor David W. Wood. He will talk about the ICAP tag technology he invented, which is self-removing affinity tag system. It can provide highly purified proteins in a single step based on a universal platform. David Wood is a professor of chemical engineering at Ohio State University and is the scientific co-founder of the company Protein Capture Science. He completed a double major in biology and chemical engineering at Caltech and then worked at Amgen on the Neupogen process in the early 90s. He completed a Ph.D. in chemical engineering at Rensselaer Polytechnic Institute and worked briefly at Bristol Meyers Squibb before starting his career in academia at Princeton University. Over the past 25 years, he has developed a self-removing tag technology with the goal of creating a disruptive downstream processing platform for the biopharma industry. Before I hand over the stage to Professor David, I would like to inform everyone that if you want to ask the questions, there's a icon at the right side. Please change it to the question mode and write there your questions, and that they will be answered after the talk. Dr. David, the stage is all yours, please.
Thank you. I'm trying to find, there they are. So first, thanks for this opportunity. I'm really happy to be here, and I'm really excited to see how many people are here seeing this talk. And I hope that I can impress upon you that this is going to be an exciting technology, or is it an exciting technology for you to be able to use, both in discovery and in full-scale manufacturing. So first, the usual disclaimer. I'm a professor at Ohio State University, but I've started a company called Protein Capture Science that is selling this technology now. We've been on the market for about 3 years. But the usual disclaimer most universities require professors with companies to give is that the university is not endorsing our product. So what I wanted to do was start with just a little bit of background on self-removing affinity tags and how we developed them several years ago and what led to this company. Our tag is based on an intine, and intines were originally discovered in the very early 1990s, between about 1990 and 1993. I spent the first 10 years of my career explaining to people what intines were, but I'm finding that more and more people are familiar with them, especially because of the New England Biolabs impact system that came out in the late 90s. Basically what an intine is it's like a self-splicing intron, but instead of splicing at the RNA level, it splices at the protein level after it's been translated. What's interesting about intines is that with some fairly trivial mutations, you can create intines that cleave on one side or the other. And in our lab and in my career developing the technology I'm going to show you today, we focused on C-terminal cleaving intines, mostly because they can be induced by fairly simple changes in the process, temperature, pH, whereas N-terminal cleaving requires high concentrations of thiol compounds, which are usually incompatible with a lot of proteins and a lot of processes. So all of the intines I'm going to show you today are C-terminally cleaving. And so when I was in graduate school, we did some genetic selection. We identified a fast-cleaving intine mutant, and we put an affinity tag on one end of the intine, and we had the protein of interest on the other end of the intine. This is sort of a funny poster. I made this slide when I was in graduate school. It's probably 30 years old now and was made on software that no longer exists, but for nostalgic reasons, I always keep it. But the idea is that by putting this self-cleaving intine between the affinity tag and the target protein, you can make a tripartite precursor protein bind it to an affinity resin, and then cleave with a simple pH shift, where the intine actually mediates the cleaving. You don't have to add any peptides or proteases or metals or anything exotic. You can literally do this in phosphate buffered saline. The intine mediates the cleaving reaction, the tag stays on the resin, and you get this really nicely purified protein. And you know, this is an example of one of the many proteins that we purified. It's acidic fibroblast growth factor. Up in lane 1 and 2, you can see the uncleaved precursor protein. 3 and 4 is the flow through, where you see that it's binding to the column and not flowing through. And then the acidic fibroblast growth factor is here down at the bottom. It's cleaved off. And then in the last few lanes, you can see we're regenerating the column. This is maltose binding protein, so we just add maltose buffer. And now the cleaved tag comes out. You can see that it hasn't completely cleaved. There's still a little bit of tag protein that comes off, but most of the protein is cleaved. And so this was really exciting. My first paper as a graduate student was in Nature Biotechnology. It was a lot of attention was paid to it. And about the same time, New England Biolabs created their impact system. They had a lot of patents that prevented us from going anywhere with our technology. And so for a lot of years, I focused on sort of the tag and kind of democratizing this. I went to a lot of conferences, but the first thing everybody would ask is, you know, what happens if you do this with a glycoprotein? And the problem with the glycoprotein is that because the intine is intact, the intine can start cleaving really at any time as soon as it's made. If you're making proteins in E. coli, you can adjust the temperature, and the intracellular pH of E. coli is about 7.5. The cleaving half-life is pretty long, but the expression times are relatively short. On the other hand, if you're in CHO and you're closer to 37 degrees C and the growth media is closer to neutral, its cleaving half-life is pretty fast, maybe 1.5 hours. And as a result, if you make proteins in CHO and they're secreted into the growth media, you won't be able to do any work. And honestly, even in E. coli, I mean, the tag will basically cleave before you can purify anything. Even in E. coli, some proteins cleave faster and slower than others, and it led to a lot of problems with reproducibility. And so a few years ago, we were on a DARPA project, and the goal of the project was to create a suitcase or briefcase-sized device that would allow you to produce any biopharmaceutical in 24 hours. So if you were a soldier in the field and you needed some insulin or Humira or growth hormone, you just type in that request, and this device would provide that drug. And I was included on a team where the intine would be the base technology. And what ended up happening is we ran into that self-cleaving problem, premature cleaving. And so I had a student from China named Changhua Shi, who's a co-inventor on a lot of our patents. And he said, listen, what we need to do is go to a split intine strategy, where instead of having the whole protein made with the tag on it, what we'll do is we'll put half the intine with the tag, the other half with the target protein, and until those two intines find each other and reassemble, it can't cleave. And that's how we're going to solve this problem. And I pushed back, but he convinced me. So we got the NPU DNAE intine, and we kind of got to work on it using everything that we knew about intines that we've been figuring out for the last 20 years. So we moved the affinity tag to the other end of the N intine. And the reason for that is that it has to do with the intine cleaving mechanism being regulated. We added our favorite D to G mutation that accelerated cleaving in the delta ICM, our original contiguous intine. And we silenced a couple of cysteine residues to decrease the sensitivity for thiol. And eventually, we changed the serine to histidine, which increases the pH sensitivity. But at the end of all of this, we had an intine that wasn't very pH sensitive. And so we started focusing on the N terminus of the N intine segment, and we created a sensitivity enhancing domain that basically greatly enhanced the pH sensitivity of this intine. And the result is that you have an intine that at pH 8.5 over 16 hours cleaves very little, but at pH 6.2 over even 5 hours, it's almost completely cleaved. So this allows you to load large volumes onto small columns and do a fairly long loading and washing process without losing much of your target protein. But then when it's time to recover your target protein, you can sort of trivially change the pH and have a fairly fast release of your protein from the column. And so then we took all of this development work that we've done, we covalently immobilized it onto a resin backbone, and that became the product that we now sell. But what I wanted to show you, just for historical reasons, this was the very first column that we poured with the prototype intine and the very first protein that we purified with no optimization. It's green fluorescent protein. Here's the tag protein, the flow through. We overloaded the column, but now the completely tagless native protein is eluted at very, very high purity. And this was literally the first time we tried it out in a column, and it worked really, really well. So we went ahead and did some additional development work that I'll explain briefly in a minute. But we ended up with what we call the ICAP tag, which is short for intine capture tag. So the idea is that it captures your protein, it uses an intine, and then it self-cleaves your protein. And so the idea is that you would buy the resin from us. And I think now is an important time for me to mention that the manufacturer of our resin is Repligen. So they are a very reliable and well-recognized manufacturer of resins at all kinds of scales. They produce GMP resins. They have their OPUS system, which allows fast scale up, and they're a very, very major player in this field. And I think that's important because a lot of people are nervous about adopting a new technology that they're not sure is always going to be available. But because we're engaged with Repligen, the supply chain and the quality of the resin won't be a problem at all. They have all the quality assurance that they need. And then you buy that resin from us. You don't need a license. There's no royalties. You just buy the resin the way you would any other resin, and then you use it the way you would want to. You add this 36 amino acid. Technically, it's 35, but if you put a methionine start codon on the front, it becomes 36. But you put it on the front end of your target protein. And then you express it in whatever your favorite host is. We've done a lot of work with mammalian and microbial in my lab. We have customers who've done all kinds of exotic things, even moss and fungus and plants and all kinds of weird bacteria. And it seems to be working. We haven't had any major protease problems yet. But basically, the intine is stable. It doesn't do anything until it assembles onto the resin. And so once it assembles onto the resin, these actually fold into a complete globular active domain of the intine. But again, because of the sensitivity enhancing domain, if you do this at pH 8.5, the cleaving is suppressed. And then you go ahead and you wash it, and then change the buffer pH to anywhere between 5 and 6.5, which will accelerate the cleaving. Now, and then you can go ahead, stop the flow, wait for the protein to cleave, either for a few hours or overnight. And then what elutes off the column is a very pure target protein while the tag stays behind. There's a couple of caveats that I want to put in here. One is that the control isn't digital. It isn't off or on. At pH 8.5, the cleaving is very slow. At pH 6.0, the cleaving is much faster. But if you have a protein that cannot handle, say, going below pH 7, you can do the cleaving at pH 7.3. It just takes longer. And so, and conversely, if you have a protein that can't go above pH 7.5, you can try to load and wash a little faster. You'll lose very little at 7.5 and then shift the pH. So there's some modularity in there. And I just wanted to also make that clear because I get a lot of questions about, you know, these pH changes, what if our protein can't handle it? And so the nice thing about this that allows you to go clear through manufacturing scale is then you can do an acid wash or even a urea or a guanidine wash and go ahead and remove the tag and regenerate the resin and use it for additional cycles. The tag really likes to refold. So we've had one customer boil it in SDS to get the tag off completely, and they found that it still worked. So how to use it? It's fairly straightforward. We have example plasmids in our on our webpage. The thing to remember is that this is a tag, like a his tag or a strep tag or any other kind of tag. So you can add it to your own favorite plasmid. And as a result, we don't sell plasmids with the tag. We just give you the sequence, and then whatever your favorite way of putting tags onto proteins you can use. And so the tag sequence is right here. It's in the brochure on our webpage. You don't have to frantically write it down. It's public domain. And again, we don't license or charge royalties on this. We just want to sell you the resin. You have your favorite way of putting tags on proteins. I know if you're in industry, you tend to just have these things synthesized. If you're in academia, my students use Gibson assembly. That's their favorite thing to do. Now you'll notice that the first 3 amino acids of the protein of interest are called out, X plus 1 plus 2 plus 3. And that's because those first 3 amino acids can impact the cleaving rate once you change the pH. And so, but I'm going to get to that right now. We've done a lot of work looking at those first 2 amino acids. And so this is sort of a limited factorial experiment. In this graph, each one of these graphs, the first amino acid is the same. So all of these cleaving reactions in the upper left, they all start with phenylalanine. And then the second amino acid, each line in the graph is a different second amino acid. So what you see in the upper left, if you have an aromatic residue at the beginning of your target, it's going to cleave pretty well no matter what. In the upper right, you see that if you have proline as the first amino acid of your target protein, it's not going to cleave at all. Proline completely abolishes cleaving. But for the other amino acids that we've done, and we sort of took from families, what you see is that the second amino acid can have a pretty strong influence on the cleaving rate as well. But what we realized is that the second amino acid's impact is very similar to the first amino acid's impact, and they're sort of additive. And that allows you to have a pretty predictive idea of how well your protein's going to cleave. But more importantly, if your protein doesn't cleave well, it gives you a pretty clear roadmap of how to get it to cleave well. So what we would say is if you have a protein that you have flexibility on, if you change the second amino acid to an aromatic, phenylalanine or tyrosine, what you'll find is that the cleaving can be greatly accelerated. And so this allowed us, and this again is on our webpage, you can use it, this sort of graph of first amino acid in the x-axis, second amino acid in the y-axis, and then sort of a rough idea of how your protein's going to cleave. We haven't done every possible combination, although honestly, I do need to update this because we have a lot more examples. And one thing that I do want to tell you is we have a new prototype resin now that we're experimenting with that will greatly expand the size of the green zone. It seems to work with a much broader range of amino acids and work well. The blue cleaves so fast that it'll cleave while you're loading the column. It'll cleave fast even at 8.5. The green will cleave in a few hours at room temperature. The orange will cleave overnight at room temperature. And then red is where we have problems with cleaving. And of course, there's proline that abolishes cleaving. What's really nice about this system, though, is that when you tag your protein, and bind it, you can see that it binds. And then what we do is we bind and wash in a bulk media, in a bulk resin. And then after you change the pH, you can take a wide barrel pipette and take samples of the actual resin, boil those and run them directly onto a protein gel, and actually watch the uncleaved protein disappear over time. This is hours up above, and watch the cleaved protein build up. And then you throw that resin into a regular gravity column and you can elute the protein. And so what's beautiful about this experiment is in a single day, you can find out, is your protein expressing solubly? Is it binding? Is it cleaving? How fast is it cleaving? Once it's cleaved, is it eluting and not aggregating on the resin? And then you can regenerate the resin and see sort of how complete the cleaving went. Although by the 5th hour, you can see it's pretty far along. And so it's a really fast, easy way to optimize proteins. And it turns out you can do this sort of in high throughput. You only need a few hundred microliters or a couple hundred microliters of resin to do this experiment, which means you can also do this experiment with a bunch of different buffers and excipients. And it turns out our Intine really, really is flexible about that kind of stuff. And so you can add urea, 4 molar urea, and it'll cleave. You can add 5% detergents, and it will, you can wash it and cleave it. You can add 2 molar salt, and you can wash it and cleave it. And the Intine doesn't care. It's happy with all of these things. So you can really do a lot of optimization, and I'll show you a couple of examples. So here was a few easy examples. Ironically, one of our easy example was the COVID receptor binding domain that does not express well and is hard to purify, a molecule that needs no introduction. It starts with the amino acids T and L, which we thought were going to cleave pretty slowly. The ones to the left cleave fast, the ones to the right cleave slow. But when we did our gravity column experiment, what we saw is that it actually cleaved almost to completion in 3 hours and had no premature cleaving at the high pH during the wash. And then it eluted really beautifully. So after we got done with the gravity column, we put it into an ACTA, just a regular high trap column. And sure enough, we get a really nice eluted band. The gel didn't stain evenly, but the mass spec sort of speaks for itself. So here's the single ion peak, the double ion peak, the triple ion peak. And what you don't see is really a whole lot of contaminants. But you also don't see any uncleaved material, which would come down, right about here, just above the molecular weight of the single ion peak. And so it shows that the cleavage was complete and the protein is extremely pure. Another example was a single chain antibody that we did. Again, it's involved in diagnosis for, I think, triple negative breast cancer. It's called ERBB2 antibody. We thought that it would cleave slowly, and in this case, it was correct. It took basically overnight to cleave most of the way to completion, but it eluted nicely, and we did a PNGase assay and showed that it was not glycosylated, which we didn't expect it to be. And then we ran this through our high trap column, and we went from this cell culture supernatant. You can see the expressed protein in this case, but a really nicely purified protein on a silver stain. And here's the SEC-HPLC, where we see some buffer artifact, but really no sign of aggregate, a very clean single band, and not a lot of impurities. And again, the mass spec, single ion peak, double ion peak, triple ion peak. There might be an impurity down here, maybe some non-covalent dimer, but again, no evidence of uncleaved protein and really not a lot of clear impurities. It's very consistent with the silver stain gel. So one of the things that we've started experimenting with that I think may be more interesting to people who at the research scale is, what do you do with difficult and unstable proteins? And so in that case, when you first make your clone, you can actually add additional tags and solubility domains to the front end of the ICAP tag and express those. And what's nice is that when you bind and cleave, all of those tags and additional affinity proteins and solubility domains remain bound to the cleaved ICAP tag, and you still get your target protein with nothing on it. And so one of the first things that we tried was this 2-domain EPRS. My student, Sabat Gonzalez Serrano, took care of this. He went a little crazy in my mind. He put a his tag, then a strep tag, then sumo, then maltose binding protein, and then our ICAP tag. We call it NPUC in my lab. And then he put it on this 170 kilodalton protein that he expressed in the cytoplasm of HEC293. And this protein is complicated. It's got a linker and 2 active domains, and it's very, very hard to make because it just does not like to fold, even in mammalian cells. But sure enough, he was able to get a really nice purification, completely native and active protein, and we were the first lab in the world that was able to do this. Another one, I had a rotation student for one summer, Katerina Kremer, or Katya, and she was looking at interferon alpha 2b. It's very prone to aggregation. And so what happened was, when we would do the purification, we'd see great cleaving. So the precursor would disappear, the cleaved protein would appear, but then in the elution, we didn't see anything. And when we regenerated the resin, sure enough, all of the protein had aggregated on the resin. So she took a bunch of little gravity columns at 200 microliter scale and just ran a bunch of different buffers. So this was our control. This was CHAPS. Again, the cleaving reaction was great, but it didn't help solubility. This was half molar sorbitol. Again, no problem with cleaving, but it didn't improve solubility. And then finally, we have a winner. She put in 0.6 molar arginine, and perhaps unsurprisingly, the cleaved protein shifts from the regeneration to the elution. And again, nicely cleaned. And then to kind of show how easy this is, this was a high school intern who was just there for the summer. And he managed to purify a membrane protein using tergitol as the stabilizer, maltose binding domain, and sumo as the solubility domain, and then our purification. And on the left is a control to show that the tergitol didn't interfere with cleaving, and of course it didn't. But on the right, what you can see, if you can follow my pointer, that's the eluted, now soluble and active membrane protein. And so this is a high school student in one summer made an active membrane protein, and we're getting ready to purify this now. And so we got to the glossy brochure, and I'm wrapping up a little bit, but I think this is going to be a very useful technology for people who are discovering new proteins. But what's really nice about it is we've designed the resin to be high binding capacity, simple buffers, and reusable, so you can go to full-scale manufacturing with this process. And the goal is to reach the early discovery groups who are finding the new drugs, have them build into those new drugs that they will cleave really well and really efficiently with this system. And then as that goes into animal models, early characterization, clinical trials, and eventually full-scale manufacturing, they don't have to change the purification process effectively at all. And so if the way things are now, you put a His tag, use a protease, your mouse cancer gets cured, and then you throw it over the fence to the human patient, you find out that actually it's really, really hard to purify without that His tag. The ICAP tag makes that go away. Now you can get the tagless protein without a protease and really without anything complicated. And you can optimize. We've even started cleaving proteins directly into formulation buffers. But what I did want to say, because the reason I'm here at Cube Biotech giving this seminar today is we started talking to them about some of their magnetic blanks. And I can tell you, for the first time being announced here at this conference, at this talk, coming soon, the ICAP mag, which is our magnetic bead. So it's the same thing. We've put our half intine covalently bound to a mag bead backbone. Target protein comes in, binds, you wash it just like you would with the mag bead. Then you drop to 6.2 at room temperature, and your protein comes off. And this is one of my favorite gels. I mean, obviously the purification looks awesome. But at 0 hour, the resin is totally green. The mag beads are totally green. And then at 1, 3, 5, and overnight, the protein just sort of falls off of the resin, and now it's completely cleaved. And then after elution, the beads are back to black. And what's nice about them is that because our intine is so robust at refolding, you can regenerate these with guanidine, completely strip off the tag. But because it's covalently bound and these are mag beads, they're fine with guanidine, and our ligand snaps right back and it's ready to go. And so at this point, I just wanted to point out, so Sabat is a true genius in my lab. He's got 1 year before he graduates. If you guys are looking for incredibly smart and capable people, you should really be ready for him. And Sophia and Nathan have also done a lot of the work that you saw. Brian's actually at Eli Lilly now. And then I have a whole bunch of undergraduate researchers who have done a lot of the work that you saw in this talk. You can see where some of them have ended up. Jordan's going to be an exceptional. She was doing an internship at Merck, and now she's doing an internship at AbbVie. But she's going to graduate in about another year, and she's another extraordinarily strong person in the lab. So I see there's a few questions in the chat, so I'll start with those. But go ahead and keep asking the questions. So yeah, so the first question, it has to be on the N-terminus, right? The position of the ICAP tag. Yes, that is true. The cleaving mechanism involves an asparagine residue at the end of the intine that cyclizes. It attacks its own downstream peptide bond and leaves a succinimide ring. And as it does that, it cleaves the peptide bond and leaves a native N-terminus. But you cannot put the intine on the other end. You theoretically can, but then you're looking at a different cleaving mechanism, and that's the one that involves thiol. That was the New England Biolab's impact system, and it's not practical for a lot of reasons. So does the incubation on the column need to be at room temperature, or could you conduct it at 4 degrees? So, so we have had mixed results. So we have definitely had proteins that we've tried at 4 degrees, and they did not cleave at all. Even over a few days, they didn't cleave. We've had other examples where we'll put aromatic residues on the front of the protein and it'll cleave really fast, faster than you would believe at 4 degrees. And so we're still trying to figure out exactly what the differences are and if they can be rescued with the aromatics. But at this point, it's something that I'd have to say it seems to vary by protein. And we definitely have some that cleave, definitely have some that don't. And it would be something that you would have to try out. As a platform at 4 degrees, I'm still very hesitant to push it, though, because I do think it has a much higher rate of having really slow cleaving at 4 degrees. But we have had some success there. Is it possible to extend the N-terminal tag at its N-terminus to affect diffusion? Yeah, again, I think you probably asked that question before I got to all of those tough protein examples. But yeah, you can add all kinds of stuff to the front end of this. We've had people who've added green fluorescent protein to both track cleaving and regeneration of the column. We've had, in my lab, my students put Sumo MVP on everything now just because it seems to be really easy. So they a lot of times don't even check to see if it's soluble. And we've been a little bit imaginative with, we took a heparin binding domain and we put it on the front of the intine, and it turns out the heparin binding domain binds very tightly and specifically to DEAE resin, almost like an affinity tag. You can run a first purification with the heparin binding domain on a DEAE resin, and then follow that onto the intine resin, where you might have to change the pH, but you don't have to really do any buffer exchange because the intine will bind in high salt. You bind to the intine resin, and then you get a secondary purification and you cleave the protein. So you can really kind of get imaginative and think out of the box about what kinds of things you would want to put on the front of the intine. But it's been really nice for that. Almost everything we've ever tried has succeeded. I don't know of anything off the top of my head that failed. And we've put some pretty crazy things on the front of this intine. Are there any typical buffer compounds? Oh, sorry, the loaded question. Well, okay, I'll start. Have you seen the influence of the tag sequence on the target's expression, folding, or activity? So anecdotally, in E. coli, the tag seems to actually improve expression for cytoplasmic expression in microbial hosts. The tag itself is charged and completely unstructured, so it almost acts maybe like a folding enhancer. In mammalian cells, we haven't been able to do those experiments very well, largely because the secretion signal seems to need to be matched for whatever's after it. So if you take, say, EPO, which is one of the case studies I haven't shown, it'll have a tag that's great for EPO, but then we want to put a tag on that's great for the intine, and they may be different secretion tags. And so the result is it's hard to directly compare them. And then, of course, the other big problem is that a lot of these mammalian proteins that are well characterized, they've created stable cell lines, they have fed batch processes, they have really great expression, it's highly optimized. While we're in an academic lab, everything we're doing is transient transfection in a shake flask. And so we haven't been able to do really clear quantitative comparisons for secreted proteins in mammalian cells. But I do keep getting word back from people working in microbial expression that the tag actually seems to help significantly. How does this compare to protein select from Cytiva? So the first clear difference between them is the controllability of the intine. So our intine at pH 8.5, it's very, very slow. Below pH 7, especially getting toward pH 6, it's much, much faster. Whereas the Cytiva resin, the protein select resin, cleaves, at least they say, consistently at all pHs between, I think, 6 and 9. And the result is that as you're loading and washing your column with the Cytiva resin, the protein immediately starts to cleave as soon as it binds, whereas with ours, you can stop that. Now, what Cytiva has said is that if you can bind and wash in 30 minutes, you can minimize your losses, and then you incubate for 5 hours to recover the protein. I think at laboratory scale, that's probably useful. But as you scale up to manufacturing scales, at any significant scale, you're not going to be able to bind and wash in 30 minutes. And And so the way I see it, you know, I realize we're competitors, but I'm going to put out an olive branch and suggest that the Cytiva resin may actually have an advantage for pure research, where you may have proteins that are very sensitive to pH and you really want to keep that pH constant, and you're generally working at smaller scales. They may have a real advantage there. Whereas if you go to large scale and you're making a drug, that drug's probably going to have to show some pretty good stability across a set of conditions, because it has to go through all of its processing and then it gets formulated and injected into people. And so if you have a fairly stable molecule that's not really that sensitive to pH and you need to make it in very large amounts, I think that our resin's going to have a clearer advantage in that case. In terms of the yields, the recoveries, I don't know if there's enough data yet from the Cytiva resin for me to make a clear comparison. I know that in their marketing materials, they've made the claim that it's completely agnostic to the target protein, but then they also have a similar table to ours of which amino acids cleave faster and slower. I can tell you both are the NPU DNA entein, so we use the same molecule, and I expect there to be very similar behavior in terms of the target proteins, but I haven't seen a lot of data on that. So I think the main difference is the ability to control our entein, whereas their entein seems to cleave well at whatever pH it's at. And then they have, you know, a 4 molar urea with sodium hydroxide to regenerate the resin. We're using phosphoric acid. But the truth is, this entein, I think probably theirs and ours, really like to refold. So if you wanted to use 8 molar guanidine, it would be completely fine. And so I think that both resins are going to have a home in the world, and I'm hoping that ours gets used for scale up. Yeah, it's a nervous question. So are there any typical buffer components that the ICAP beads are vulnerable to? So the only thing that we've seen that's sort of in the literature is divalent metals at fairly high concentrations will inhibit the cleaving. Zinc especially is one of the ones that's known to inhibit cleaving. Anecdotally, when we've tried to use a his tag followed by our entein purification, we found that imidazole seems to interfere with the cleaving reaction at a fairly high concentration, but we haven't done any really quantitative evaluation of that. Outside of that, we've used a huge variety of different buffer compounds. Tris, bistrus, bistrus propane, AMPD, pipes, MES, phosphate buffered saline, citrate, glycine, you name it, we've used it. And we've used arginine as an excipient, aspartic acid as an excipient. We've used different detergents to solubilize membrane proteins. We've used 2 molar salt washes. We've used 5% tween and Triton washes. We've had people who've said they can bind actually in 4 molar urea because they were using our entein as part of a refolding process. And so the short answer is no, with maybe a couple of exceptions of high concentrations above 10 millimolar divalent metals or anecdotally imidazole. But I would say use it with imidazole. If you see something weird, it might be the imidazole, but we haven't really proven it conclusively. How does it work for small peptides? So we have not done a lot of work with small peptides just because in an academic lab, it's harder to characterize them because we do a lot of our characterization on Kumasi gels. And small peptides, as you probably know, don't always stain very well, and they may not transfer well or stick well if you're trying to do a Western. And we don't have the kind of HPLC and mass spec easy in our lab to really look at it. The worry that we've had is that because the entein tag is unstructured and the small peptide's probably unstructured, that you may end up with a protease magnet, where a protease is going to really love seeing this completely unstructured peptide. But if you put a maltose binding tag on the front of it, it might protect it. And so we would be really happy to work with you if you want to try it with small peptides and help you troubleshoot. We don't know of any reason why it wouldn't work with small peptides, unless there's some weird structural thing that happens. But yeah, I think it should work. We just haven't had the equipment that we need to really explore that very well. So have you tried protein expression in yeast? There will be a signal peptide. There might be a few amino acids left upstream of the tag. Will it work fine with the binding of the tag to the resin? So we have not tried it in yeast. When I was first doing my first peptide, intine, we did do some work in Pichia, but we just did not get very good expression. And I became very cynical about Pichia. So sorry about that. But we do have other people who are looking at it in yeast. So if there's a few amino acids upstream of the tag, that's totally fine. You can put a whole maltose binding protein upstream of the tag. And in fact, if one of the things that we're experimenting with now, and we've seen some other customers experimenting with, is they'll take actually a really well secreted domain, put the signal on that, put that on the front of the intine, and that domain and the intine will drag their favorite protein out into secretion. That's worked sometimes really well, sometimes not as well, but there's a lot of things that you can do. But a few amino acids upstream of the tag is absolutely no problem at all. We've put the kitchen sink on the front of the tag, and it seems to bind and cleave just fine. The second issue is that the optimal pH is pH 6s, so the secreted protein. Yeah, so that's not a problem because, again, because it's a split intine and you only have part of the intine, it can't cleave until it sees that resin. And so what we've done in mammalian expression where it's secreted is we'll take the cell culture supernatant and literally titrate it with sodium hydroxide up to pH 8.5 before we load it onto the resin. And believe it or not, we don't have any problems with that. We'll filter it if there's any aggregates that form when we titrate it. But that's actually worked really well. And I have a whole set of slides where my student did a study and used really dilute sodium hydroxide. And what he eventually found is you could add pretty concentrated sodium hydroxide, obviously slowly and dropwise, and you titrate it. But yeah, you can adjust the pH. And then once it's at pH 8.5, you dump it onto the resin, it binds, but by then you're now in control of the pH. So at that point, you can adjust the pH to whatever you need to get the kind of control that you want. So yeah, in yeast production, it's not a problem at all as long as you don't bind at pH 6. If you're binding at pH 6, then you're going to have a problem. So you would just secrete the supernatant. The protein yield per mg of resin. Oh yeah, sorry, I forgot to mention that. So the thing to remember with the resin is that it's a stoichiometric binding. So if you have a large protein, you're going to have a larger mass loading just because each monomer has a higher mass. So when people talk about protein A, they talk about grams per liter, but all monoclonal antibodies are the same size. So it doesn't matter. So at that point, grams per liter makes sense because all of your target proteins are the same. With our resin, I showed you 140 kilodalton protein that we purified, but we've also purified Neupogen, filgrastim, which is 12 kilodaltons. And as you might imagine, if you have stoichiometric binding, the mass loading of those two targets is going to be pretty different. So that's the big caveat for the answer. But what I can give you as a benchmark answer is we have streptokinase as sort of our favorite test protein. It has a molecular weight of 55 kilodaltons, and we can recover 40 grams per liter of that protein from our resin. So the density of the ligand is about 1 millimole per liter, which means that if you have a 15 kilodalton protein, you should be able to bind 15 grams per liter. Now, obviously, with dynamic binding capacity, you never get 100%. So I would say you would get about 70% of 1 millimole per liter. So 15 grams, 15 kilodalton molecular weight, 15 grams per liter. 40 kilodalton molecular weight, 40 grams per liter. Streptokinase, 55 kilodalton molecular weight. It should bind 55 grams per liter, but we usually only get about 40 grams per liter. There's obviously going to be a steric thing at the very high end. And so, but from a molar binding capacity, it's about 3 times higher than a good protein A resin. So I know there's a lot of caveats to that. Regarding to the purity, have you tested for host cell or nucleic acid contamination? So beyond the MALDI and the Kumasi gels and the mass spec, we have not done a host cell protein ELISA. And the reason is because those are expensive, is the main reason. But we have done a qubit assay for nucleic acid. And in all of the examples that we had on mammalian proteins, the nucleic acid, the DNA that was left was below the limit of detection. And so we seem to be able to remove nucleic acids quite well. And we think the host cell proteins are all gone, but the gold standard is obviously that HCP ELISA. We've done some LCMS stuff, but the purity is quite high. And so I wish I had more data to show you, but that's all I can tell you. Is the qubit assay, it was below limit of detection. And we did a before and after, and we did see greater than 4 logs of reduction in host cell DNA. But we don't know what the actual final concentration was. And then the maximum number of times the resin can be reused. We've done simulated regeneration up to 50 cycles, and we didn't see a loss of binding capacity. The big warning I have is that we realized later we weren't saturating the resin every single time. So we might have lost some binding capacity, but just not seen it because we weren't saturating the resin. I've asked my students, realistically, if you take the column that you've used the most times to actually purify a protein, how many times is the most that we've used a single column? And they said that they know they have one column where they have purified 18 proteins on it. Maybe the same protein a few times, but it's been used for a bunch of different proteins and has done 18 full cycles of purification and regeneration with a legitimate protein, and they're still using that resin. And so we think that it's going to be 50 per 50 at least, and maybe 100. How efficient is the tag cleaving in percentage, not purity? So the cleaving reaction behaves like an exponential decay because it's a molecule that's cleaving. So the longer you go, the more cleaving you get. We do not see on the resin, we don't see proteins that only cleave partway and stop. We had seen that with the contiguous intine, but we don't see it with this split intine. And so if you have a slow cleaving protein and you don't want to cleave it for very long, then you're going to lose a lot. But if you have a fast cleaving protein or you're willing to wait and your protein's stable, you can get sort of arbitrarily high percentage of cleaving and really recover, really close the mass balance. You know, the protein you see in your supernate and going onto the resin all comes off. And so, so there you go.
Thank you so much, David. I think the questions will keep on coming up. And as a scientist, protein scientist, I can say it's really nice technology you've developed. All the questions can be sent to us via email to Cube Biotech, and then we can forward it to you, and then we can answer them, definitely.
Yeah, and I suppose I should say that this is the name of the company is Protein Capture Science. Yes. And our webpage is proteincapturescience, all one word.com. And you can order direct. We have an online marketplace. We have bulk resin. We have 1 mil and 5 mil high traps. We have robo columns, and we have what we call a troubleshooting kit that I think we should rename optimization kit, where we give you a little bulk resin so that you can do that time course experiment, and then a couple of columns so you can go ahead and do high purity work, all in one kit. And then coming soon, ICAP Mag. Which I think is going to be a lot of fun for people to use as well.
Yeah, I hope this will be nice.
Yeah, and we have working prototypes where we use the Cube Biotech magnetic cores. Perfect. And they look beautiful. They have really high binding capacity. So it's been great to work with you guys.
Nice, perfect. I would again like to thank you for the talk. It was really nice, and thank you for all the audience. And please stay tuned for our next masterclass on 10th of July, and details will be sent by email. So have a nice evening.
Thank you.
Bye.